All right. Well, welcome to Wave Club, everybody. It is 9:31, and we're going to get started. Um, I am really happy to have you here today for our rescheduled event from last week at this time. Sorry about those issues last week. Our presenter today, anyway, my name is Josh Jacobs, for anybody who hasn't been at Wave Club before. My partners in Wave Club here are Bard Ehrmantraut, Uma Mohan, and Irfan Zabeh, whose faces you can see on the right-hand side there also. Uh, Irfan and Uma, thank you so much for helping to organize everything today. And I'm really excited to have our speaker here today, Vishnu Sreekumar, who is staying up late for us. Vishnu is in India now, so it's a little late. Vishnu's done a lot of cool work on traveling waves, that he's going to tell us about today. So, Vishnu, thank you for coming. Vishnu is at the International Institute of Information Technology in India. He's going to give a talk today on From Traveling Waves to Spikes, a multi-scale view of neural communication. Um, and we are going to post this recording of this talk on YouTube for anybody who's interested. I hope that's a useful resource for everybody. Um, that's what Vishnu looks like in the sunshine with his sunglasses on. Here's our YouTube link. You can just go to @Wave Club Seminar on YouTube or on Blue Sky. And I think we are also on Twitter also, if that's your style, where you can go and see our previous Wave Club talks. And learn and follow us on Blue Sky to learn about what's going on. The plan for Wave Club, as in previous weeks and months, has been that we aim for a relatively brief presentation, and then we want to use of about 40 minutes, and then we want to have time for the presenter to interact with the audience, and we want to say 15 or 20 minutes for conversations later. Um, I think the Q&A feature is not working today, but if you have questions, please type them in the chat window, and Uma and Irfan will help us kind of coordinate this in the end. If it's a small clarification, please feel free to jump in and we can address that while Vishnu is talking. But and he will be kind of running the show. But for bigger kinds of conversations and questions, which I want to have bigger conversations and questions, but the best thing would be to have that at the end after Vishnu's talk. As a reminder, each month of Wave Club, we have different speakers. We try to keep the Wave Club meeting as diverse as possible to talk about the general theme of Wave Club, of course, is to talk about traveling waves in the brain across all species and both simple animals and complex animals and computational models, all different areas of work related to traveling waves in the cortex. With that in mind, we have a range of different speakers, and we're now looking at our speakers for 2026 as well. These are our speakers. I will introduce Vishnu in a second more. Our speakers for the fall look like this, with Pulan Gong giving a talk in November, Mark Kramer from BU giving a talk on traveling waves in December, and then January 6th, Jun Young Moon will be giving a talk as well. So those are our upcoming speakers. But we're looking for speakers for 2026. So please, if you have suggestions for us or are interested yourself, please just come talk to us. The goal of this is to have it be a really open and fun and free-flowing conversation about different areas of traveling waves. So our speaker today is Vishnu Sreekumar from IIIT in Hyderabad. Vishnu did a postdoctoral fellowship at NINDS with Karim Zaghloul, who's a surgeon at the NINDS, and that's where I got to know Vishnu as well. Previously, he did a master's and a PhD from psychology in cognitive psychology from Ohio State University. And he also has a master's degree in physics from the Indian Institute of Technology in 2009. Um, and um, and I think I'm just going to turn over the screen to Vishnu there. So Vishnu, take it away, please. I'll stop sharing my screen. Um,
All right.
Thank you everybody for coming. It's great to have you here today, and thanks for your flexibility with the rearrangement from last week.
All right. Thanks a lot, Josh, for the introduction. Happy to be here. So today I'll talk about traveling waves and how we move from traveling waves at multiple different spatial scales to spiking. Um, but before we start, I would like to set up some context. So, like Josh mentioned, we are a lab that's set up at the International Institute of Information Technology in Hyderabad, India, which is a tech institute that is mostly known for its strengths in AI and ML. And as you can see here, this is our lab. Our research is primarily driven by very talented undergrads, some masters by research students, as well as PhD students. So in the last 4 years of setting up a lab, we've been focused mostly at the intersection of memory, computer vision, NLP, kind of playing to the strengths and talents that we have in the local context here. And of course, funding for basic science is an issue all over the world, but especially so in India. But recently, we've been fortunate to get a couple of grants, which will enable us to set up a high-density EEG facility here. And so this seems like a good time to really start thinking once again, about building a neuroscience program here. And so I was very grateful to get this invitation from Uma, Irfan, Josh, and Bart to talk about traveling waves here, because it really gave me an opportunity to, cancel meetings and sit, analyze more data so that I could update the results that some of you may already have seen are in existence since 2021. But it's only recently that I really got to try and do some additional analyses to try and make sense of the various collection of, it was basically a collection of facts about traveling waves at multiple scales that I reported in that preprint. But today I would like to present some analyses that will suggest some mechanisms underlying how these waves at multiple spatial scales come to be related to each other. And I would love some feedback from the community before we submit this paper back again for publication. And I know that Josh keeps talking about this as an informal seminar, and I've watched all of the preceding talks. They've all been, really great talks of published work. I would really appreciate some feedback on this unpublished work. And so let's start with looking at this picture. And you'll see that this is a collection of tens of thousands of neurons collected from mice that did this no-go task, right? And you'll see that this randomly looking display of neurons coming on and off, to enthusiasts of traveling waves, they may not, look all that random, right? And I did a control F on this paper looking for propagating waves, traveling waves, but I found no mention of that. But they did some really clever analyses and showed that right after the sensory stimulus comes on the screen, you see an increase in co-fluctuations of neurons across these multiple visual cortical regions that they tracked. And so one question that arises here is that what are the mechanisms underlying such coordination of spiking activity across multiple cortical regions? So any such discussion should start with a discussion on oscillations, right? So oscillations are this reflective of this emergent push and pull between excitation and inhibition in a local population of neurons. And so naturally, because oscillations indicate these rhythmic bouts of excitation and inhibition, you see that spikes preferentially fire at the excitable phases of these oscillations. Now, why does this matter? Because we have some popular theories of how communication happens in the brain. So particularly if you look at communication through coherence, this suggests that let's assume that this is the sending site. And so there are some units that spike preferentially at the excitable phases of the local oscillations at that site. And if you look at a receiving site, these spikes better arrive at the receiving site when the receiving site is also at an excitable phase. And so this suggests that you should have these coherent and synchronous oscillations across sites to enable communication. So consider this pair of sending and receiving sites, for example. So when neurons from this site arrive at this site, if it happens so that this site is not at an excitable phase, you don't get communication between the two sites. Now, of course, people think that it's gamma oscillations that underlie this sort of communication, especially in visual cognition and in the visual cortex, because you see these very nice gamma oscillations that come about even during naturalistic visual tasks. But there are some suggestions that when you think about long-range communication in the brain, perhaps something like alpha is a better channel. For example, this paper from Karim's lab actually in 2018 suggested that you look at these intracranial electrodes. So you look at these subdural electrodes implanted in epilepsy patients. You see these very coherent oscillations in the alpha range. But additionally, what they did is they drew inspiration from some pretty old information theory and said that for communication to happen, it's not just that you need coherent oscillations at some center frequency, but you should also observe correlations in the broadband amplitudes. So what they did is they derived time delays based on the cross spectra, and they also derived a time delay based on directed correlations that they computed using the broadband activity. And they showed that these time delays really correspond well with each other. So what that means is that, so again, if you have an oscillation here at the sending site, you have spikes that preferentially are activated at the excitable phases of this oscillation. And it gets to the receiving site with some time delay. And it just so happens that the oscillation here is coherent with the oscillation here with a specific phase delay that matches the time delays that you can calculate from the conduction delays. So that sets up this ideal time window within which you get spike time dependent plasticity, right? And so what they additionally showed, like I said, is that it's not just that these oscillations need to be coherent in alpha, but you should also see these broadband correlations. And they saw that these broadband correlations were maximal if you actually looked at the troughs of these oscillations. That is, if you took broadband activity from the troughs of these oscillations, you saw the maximum correlation across pairs of electrodes. Okay, so I guess I would be preaching to the choir here if I explained what a traveling wave is, but essentially you see oscillations that propagate across the medium. It's like you throw a pebble onto the surface of a pond and you see these ripples that propagate over the medium, and something very similar happens in the brain, as we all know. But why do people think that traveling waves play a role in communication, especially this spike time dependent plasticity? Of course, Lyle Mueller has made some very interesting suggestions. So again, if you consider a sending site A and a receiving site B, if they are perfectly in synchrony with each other, it may be the case that if you have spikes that are sent from site A to site B, by the time it takes for the spikes to get here, because there's a 20 millisecond delay associated with the axons here, it is possible that the postsynaptic neuron actually fires before the pre gets to the postsynaptic site. And so this might set up conditions that are amenable more for long-term depression than LTP. And so they suggested that if oscillations are instead organized as propagating waves, what you then get is that, if you have a sending site here, the oscillation, the receiving site just happens to be at the right phase delay from the sending site, such that you get the pre before post that is ideal for SCDB. And if we believe that traveling waves underlie communication, it should also be the case that traveling waves organize spiking, because we believe that spiking is ultimately the unit of communication in the brain, right? And there are suggestions that traveling waves are potentially depolarizing. And we've seen this in modeling studies and this great review paper that Lyle Mueller and others wrote. And if you look at this picture B on the right side here, this is where they talk about this activated network that kind of mimics the aroused state, right? And so there, if you think about a current injection to a local population of neurons, you do see propagating waves of membrane potentials, but you see very sparse recruitment of neuronal spiking. And so this is what they refer to as the sparse wave hypothesis. But alpha is thought to be inhibitory, right? So even in the Shapton paper that I showed you all earlier, you find that alpha coherent units, if you look at their spike rates, they are negatively correlated with alpha power. So it is not very clear whether human alpha range waves should be depolarizing or not. And if you listen to talks by folks like Sanofsky, you will see references to waves all the way down. So it's like this infinite regress. It's turtles all the way down. It's waves all the way down, right? So it's waves built on top of waves on top of waves. And of course, we record waves from EEG, from ECOG, from Utah arrays, even at the level of units. So the question is, how do waves all the way down across scales come about? And how are they related to each other? So that's the primary question that we'll deal with in the talk today. And if you go all the way down to the level of spikes, there are some nice demonstrations that spikes can also trigger local waves, right? So this work from 2009 from the Carandini lab, they showed that if you record a spike on what is called a triggering electrode, so let's say this is a triggering electrode, you record a spike there, you see a deflection in the field, but then if you look at the other electrodes and you compute the time it takes to the negative peak of the deflection, you see that that time to peak is a function of distance from the triggering electrode, which is indicative of a propagating deflection, right? There was some debate in the literature about whether this effect was due to filtering issues, and there was a debate that happened, but I think the debate was resolved soundly in favor of this account of spikes triggering waves. Okay, so we'll keep this in mind because I think this will play a role in us interpreting some of the results that I will show you later. And finally, I also want to draw your attention to this quote from this paper from 2012, which said that waves in membrane potential can certainly influence subsequent spikes. Okay, so the questions we have are, do traveling waves provide STDP windows, which basically can be thought of as encoding opportunities? Okay, so in other words, does the presence of traveling waves indicate better cognitive processing? And then there is this cross-scale coordination question. So are macroscopic waves recorded on ECOG grids related to mesoscopic LFP waves recorded on Utah arrays? And then there's a question of whether these waves are depolarizing. Okay, can you show this using direct neuronal recordings from the human brain? And finally, what are some candidate mechanisms underlying coordination of neural propagation patterns across spatial scales? Okay, so our recordings are intracranial EEG recordings from subdural electrodes implanted with drug-resistant epilepsy patients. And most of our patients are temporal lobe epilepsy patients, so we have recordings from the lateral temporal cortex. And simultaneously, for a subset of these participants, we also have Utah arrays that are implanted in the middle temporal gyrus. So which is along the path of the overlaid ECOG grid. So we get both local field potentials as well as spiking activity from the Utah arrays. So our participants are engaged in this associative memory task. So they see pairs of words at encoding. So for example, beam and camel go together, flood and rhino go together. So there is a 600 millisecond baseline, there's a 4-second task period. And then later they are given one of the two words and they have to recall the paired word. So this is a paired associates task. And so we have 20 ECOG grid patients with 6 of them having these concurrent Utah recordings. So we start off by doing an analysis that is similar to the Shapton paper that I talked about earlier, in that we computed coherence between pairs of electrodes. So right here, I'm showing you the mean coherence across pairs of electrodes, macro electrodes within one participant. And you see a peak at 8 Hertz in this participant. And so you can repeat this kind of an analysis across all participants and compute the peaks observed. And you see that they generally lie in this high theta to low alpha sort of range. And so it's generally in the 4 to 15 Hertz range. So we apply this frequency range filter, 4 to 14 Hertz range filter, and do the Hilbert transform and get instantaneous phases. And so all subsequent analyses operate on these instantaneous phase maps. And there is a particular reason why we chose this slightly broader frequency range. Because if you recall, our primary interest is to understand the temporally precise coordination behavior across multiple scales. And so given the time frequency trade-off inherent in signal processing, we cannot demand great precision and frequency because we get things smeared in time if we did that. And I have demonstrations of this that we can go over in the discussion if there are questions about that. Okay, and I should also note that these peaks that we calculate within individuals are highly similar for both macro and micro electrode pairs. So it works out that we can use the same kind of filter to analyze waves at both scales. Okay, so once we have these phase maps, so what I'm showing you here is just a smooth version of an instantaneous phase map. We apply what's called this optic flow method, particularly the Horn-Schunck method. So essentially, we solve for the x and y components of these velocity vector fields. And since you have two unknown quantities, you have two constraints you impose. This was originally designed to track objects in videos, right? So if there's an object that moves in a video, you know that the pixel intensities of that object have to remain constant across your frames. And so that defines the brightness constancy constraint that is optimized for in the algorithm. And then you also demand that there are no huge discontinuities in the motion. So that is the second term. And the alpha here is sort of important. This is what we call the smoothness constraint. And so higher values of alpha get you smoother flows, and lower values of alpha are able to resolve more detailed flow. And if you expect large coherent kinds of activity, it is okay to use slightly larger values of alpha. And if you want to resolve like very complex patterns of activity like those that you observe on the Utah scale, you can use a smaller value of alpha. But ultimately, there's no objective way to set these values. You really have to run the algorithm, look at the movies that result, make sure that they visually make sense. The arrows follow the propagation paths of the phases that you plot in these movies. So we get these phase velocity fields, and we impose a criterion on top of these fields. So to detect planar traveling waves, what we require is across all of these electrodes, we need the directions of these phase velocity vectors to point in the same direction. So what we do is we take a vector sum over all of these vectors and divide it by the average magnitude that we calculate across all of these vectors. So this gives you a number that lies between 0 to 1, and we impose a criterion of 0.7 and say that everything above 0.7 indicates that most arrows point in the same direction. Additionally, we also demand that across time frames, we don't get directions that vary, because you can imagine that at any given time frame, you can satisfy this condition, but perhaps from time frame to time frame, you get random flips in the direction. So we demand that the directions are also pretty much in the same direction. So within 30 degrees is what we demand. And we also run this on symmetric grids, because there is this concern that if the algorithm sees more information along one dimension compared to the other, it might be biased. So we always run our analyses on symmetric grids. So in the case of the ECOG grids, even though they are 8x4 grids, we apply the algorithm on 4x4 subgrids before we stitch them together to detect our longer range waves at the macro scale. And our smoothness parameter choices lie between 0.1 to 1. These are well within the ranges recommended by previous literature as well. Okay, so this gets us to the macro scale waves, which I'd like to show first. So like I mentioned, we first detect these on these 4x4 subgrids, and I detect these on overlapping 4x4 subgrids, and then later stitch them together to detect waves that propagate all the way across. So this is one example wave. So if you track any given color here, where the color denotes instantaneous phase, you can see that it propagates, in this example, from the posterior to the anterior part of the brain. So I'll just play that again. And what we see is mostly just one sweep across. And I'm sure there'll be questions about this. I'll be happy to discuss why it's maybe just one sweep, right? But essentially, we observe transient waves. Okay, so if you take some electrodes along the propagation path, you can look at the traces along with the filtered data. You can track, let's say, the troughs of various oscillations along the path. The gray shaded regions indicate time regions where my algorithm indicates that there is a wave. And you can see these phase lags across these sites. And so the algorithm does detect these phase gradients across space. For our surrogates, we use something called the Tyler-Pritchard surrogate. So we go to the Fourier space and we shift the phase by a random amount. But importantly, we shift the phase by the same random amount across all channels such that we preserve the cross spectra. And then additionally add a small amount of random phase to each individual channel. What this does is it firstly produces data that look very much like actual ECOG signals. But additionally, if you look at the spatial cross correlations in the data, in the actual data, you see significant spatial cross correlations. Now, in the surrogates, you don't want to completely destroy these spatial cross correlations because yes, if you completely shuffle the electrodes at the individual electrodes, the data will look like real data, but you should also expect spatial relationships in the data, right? Because whether it's due to volume conduction or whatever spatial spread mechanism, the surrogates should preserve at least a little bit of that was the logic here. Whereas you destroy the phase propagation patterns. And so then you can apply the algorithm on the actual versus the surrogate data, and you'll see that you find a significantly greater number of waves per second in the real compared to the surrogate. And of course, you can separate out these distributions further by destroying the cross correlations even more by randomizing the phase even more. But this was a very conservative surrogate that we used. And like I mentioned, we had concurrent Utah recordings, and you can apply the same algorithm and you find waves at this spatial scale as well. Okay, so the first question is wave occurrence indicative of the sort of encoding windows of encoding opportunities that I mentioned earlier, right? So is it indicative of communication/cognitive processing? And I should say that in none of my previous analyses have I found anything that relates to specific conditions in the memory task. But I decided to look at this at the level of overall subjects. So what you see here on the y-axis is the overall performance at the subject level on this task. So this is the overall probability that they get the answer right in the memory task. And on the x-axis is the wave occurrence rate. That is the number of waves that we detect per second at the subject level. And we see this very robust relationship between the degree to which these waves occur and memory performance. And you also see this a relationship with reaction times. That is, if you have a greater number of waves per second, you're also generally faster at the task. And I've never seen results this robust. So I tried looking at various potential confounds. For example, there is no relationship between the wave occurrence rate and the peak alpha coherence, for example. I did find a weak relationship between wave occurrence and the number of years somebody has had epilepsy for, but it's a non-significant relationship. And there is absolutely no relationship with memory performance and the number of years you've had epilepsy for. So none of them could really explain this strong relationship that I observed. And this works just as well if you took just the incorrect trials. So this is not something that you get only from the correct trials. So in fact, if in general somebody has a greater number of waves per second, it seems to be indicative of their memory success. I also did this analysis by simply computing the proportion of time that you observe wave-like activity. And it turns out that the number of waves per second is more indicative of memory performance, even though wave rate, in terms of proportion of time spent in wave-like activation states, also shows similar patterns. Okay, so then we get to the next question. So are these waves that we detect at the two spatial scales the same phenomenon that are just captured on different arrays? So we can first look at directions. So we find that at the macro scale, we see that most subjects show very bimodal direction distributions with a very strong preference for posterior to anterior. And so if you look only at the long range waves, you again see very similar posterior to anterior preferences. And this is true across participants, and this is true for both the anterior as well as the posterior parts of the lateral temporal cortex. And of course, Josh's lab has published work. And if you look at the lateral temporal cortex there, they too observe this kind of a direction preference. Um, and if you look at the anterior subgrid, you find even greater directionality compared to the posterior subgrid. And this is a result that I don't have a very good explanation for yet, but you see that these are pretty robust across participants. And even if you look at the durations of waves that you detect on these smaller subgrids, these are consistently slightly longer on the anterior compared to the posterior. So I looked at a bunch of resources to try and understand whether there were anatomical differences between the posterior and the anterior parts of the temporal cortex. I was looking particularly for, let's say, transverse kinds of gyri, either on the surface or hidden underneath. But I couldn't really come up with a very good explanation, but there's a very robust difference that we find between the anterior and the posterior. So if there's anybody here who has better knowledge of anatomy, please chime in during the discussion. Perhaps it is the case that we have maybe a smoother anatomy in the anterior compared to the posterior. And we know that there are reflections that happen at cortical boundaries and such. So which is why I was looking for evidence of more transverse going gyri in the posterior part. Okay, the second scale is a microscale. So we can look at the directions of waves at the microscale. So what I'm showing you here are the macroscale direction distributions for one participant. And then for the same participant, you can look at the directions observed at the microscale. And you see that there is no clear relationship in the directions between the two scales. And you can look at this across participants. Again, at the microscale, you see clear directional preferences. They are mostly bimodal, but very clear preference for one direction over the other. But if you look at across all the 6 participants that we have, it is not very clear that there is a tight relationship between the direction at the macro and the micro scales. Okay, so then we can examine speeds of the waves at the 2 scales. And of course, in the literature, we see that macro scale, macroscopic waves are propagated using these white matter fibers that tend to be myelinated, so they have higher conduction speeds in general. And if you look at these mesoscopic waves, they seem to be driven more by these horizontal fibers in the superficial layers of the cortex, which are unmyelinated, so they have lower speeds. And so when we measure the speeds of our waves, we again see this difference in speeds where the microscale waves are 5 to 10 times slower than the macroscale waves, which agree with these ratios of conduction speeds at the 2 different scales. So now you might have observed that these waves tend to be a bit slower than the conduction speeds that have been estimated. And so this brings me to this anecdote that I'd like to share with you all. So while I was doing my postdoc with Kareem, Kareem came to me quite excited one day after a trip to Vegas, I believe it was. And so he was driving along the streets and he came to me and said, Vishnu, these traveling waves are like traffic lights. And so I think what he was trying to explain here was if you really think that traveling waves are mediated by synaptic processes, right? So let's say spike transmission has to happen. There are some delays involved at each of these synapses. There are some integration times involved before you can really summate and see a deflection in the local field potential. And this deflection is what you then see as this propagating wavefront. So if that's the case, we should really expect these wave speeds to be a bit slower than the conduction speeds. At least, I mean, this is my speculation at this point. Okay, so this emergent phenomenon of waves should be slower than conduction speeds. Okay, so they may not be the same exact phenomenon since they probably propagate over very different connectivity, but are they related in time? So we can look at the times of occurrence of these macro scale wave times. So what I'm indicating here are just ones anytime we detect a macro wave, and ones anytime we detect a micro scale wave, and zeros everywhere else. And you can compute cross correlations in these spike trains, so to say, but these are trains of wave occurrence times. And so if you look at these within different participants, you see that there is a peak in the cross correlation between the micro and macro scale wave times close to zero. And 5 out of 6 subjects show the effect, indicating that these waves tend to co-occur. So the next question is, why do these waves co-occur? So is it the case that there is some sort of activation phase induced by the macro scale waves? So to investigate this, we looked at the macro phase coming from the closest macro electrode relative to the Utah array. So the yellow denotes the Utah array, and this is the closest macro electrode. So anytime we detected a microwave, we got the phase of the oscillation on the closest macro electrode. And let's look at one individual participant. And you see that in the true data where you're locking the microwaves to the phase taken from the closest macro electrode from the same trial, you see that it has this preference for a certain phase. Right? So anytime you see a microwave on the Utah array, you see this phase on the macro electrode that is preferred. And you can look at the trial shuffle surrogates, you see less of a preference. You can compare the resultant vector length and you'll see that they are different. And you can look at these preferred phases across all the 6 subjects. And what you generally find is that whenever you have a microscale wave, you see a macro phase that is close to the trough of the oscillations at the macro electrode. And if you look at the surrogates, you find that this is all over the place. And I should also note that the specific phases that we find here could be influenced by the distance between the closest macro electrode and the Utah array. And also, I should also mention that our algorithm really detects a wave on the Utah array only when a wave has clearly been established on the Utah array. So if you remember the PGD threshold of 0.7, that really is a stringent threshold that can be met only when all the arrows on the array point in the same direction. So you could have a wave that is entering the array and our algorithm doesn't yet call it a wave. So there is a little bit of a time delay that is involved there in detecting the wave. Okay, so now is this activating macro phase for microwaves also an activating phase for spiking? So we again looked at spiking data from the Utah arrays, and we, similar to the previous analysis, we took the phase from the closest macro electrode, and I'm showing you the same participant as earlier. And again, you see this preference for the trough of the oscillation at the closest macro electrode. And you can compute the directionality or the phase preference directionality for the true versus the trial shuffled. In general, you'll see that most subjects show the effect, but you can also observe an even more robust effect if you look at spiking that happens during the first 10 milliseconds of the macro wave that is detected on the closest macro electrode subgrid. So you can look again at the same participants and plot these things, comparing the resultant phase preference between the true and the surrogates. And you find that there is this preferred phase of the trough of the macro oscillation when you see spiking on the Utah. Okay, so all of these suggest that there is this possibility that macro waves induce an excitatory oscillatory phase that might then induce some spiking that might be detected on the Utah. So far we've observed these directional macro and micro waves, and we found that they are temporally coordinated, even though they are likely different phenomena that travel through different connections. But this coordination is likely mediated by the excitable phase of macro oscillations, but the direction of causation cannot really be established because of the time delays involved in the detection of the waves at the 2 scales. But it is clear that it is possible that these waves may induce sparse spiking. And this is where I would like to invoke the earlier result of Newhouse and team, where they say that spiking may also trigger these local microwaves. And I would also like to draw your attention back to this quote now, which says that waves in membrane potential can certainly influence subsequent spiking. So now the question is, do microscale waves modulate spiking? because the idea is that these waves in membrane potential can probably influence further spiking. So what we do here is, again, we take the time points at which we detect a microscale wave on the Utah array, and then we call that time equals 0, and we take the spike train that is locked to wave onset, and we accumulate these spike trains across all the microwaves detected on the Utah. And so if you look at the two participants for whom we had a sufficient number of units, you can see locked to the onset of microscale waves, you see this increased likelihood of spiking. Now, I should note that you can really observe this only if you accumulate across all the waves that you detect on the Utah array and all the units that you have available. So this suggests that there is weak modulation of spiking, where if you look at any individual wave, you don't get a ton of spiking. But if you accumulate across many, many waves and units, you see this modulation, okay? Which means that this is really indicative of an increased likelihood of spiking. And this really fits in with this sparse wave regime that Mueller talked about in his 2018 review paper. Okay, so we can also look at where exactly this probability of spiking peaks after the wave onset. And so for this analysis, because we did not have a lot of units across everybody, we used a high frequency broadband proxy for firing, for multi-unit firing. And we looked at the peaks in probability of firing triggered by the LFP waves. And we find that most subjects show a clear peak at around 50 milliseconds after wave onset. And if you look at surrogates, what you find is that these, even if you do observe a peak by chance, the mean latency of that is all over the place. So whereas in the true data, you find that this peak is very tight around the 50 millisecond mark, whereas in the surrogate, it's all over the place where the variability of this peak and the latency is negative in the first place. So this suggests that the likelihood of spiking really does increase after the onset of these microscale waves. So going back to this idea of STDP, if you look at Mueller's eLife paper, so they posited that these waves really are repeatable. So in other words, if you have two electrodes that are at a certain phase/time delay with respect to each other within waves, it would really help if you can replicate the same kinds of relationships between these phases across many repeats of these waves, so that you get more opportunities to strengthen the connections between activity at these electrode sites. So what they did is they computed the similarity of the phase maps across repeats of their spindle oscillations. And they found that if you look at individual electrodes, they take on a very narrow set of phases across waves. So we too ask a similar question. So if you look at these waves detected on the microelectrode array or the Utah array, do you see consistent phase across waves moving in similar directions? So to do this, we took the central four channels. It could be any channel or any individual channel, but I chose the central four channels. And anytime we detected a wave at the Utah on the Utah array, we took the phase on these four central channels. And across all the waves, we looked at whether there was a preferred phase at any given channel. And so again, you see that only in the true data, but not the trials shuffled surrogates, you find that there is a certain preference for the phase that gets induced on each of these electrodes during these microscale waves. So what this suggests again is that these waves are repeatable, especially if you look at waves that are within the same kind of direction. Now, fortunately, because you see that you have strong directional preference, you see the same effect even if you did not do this restriction to the main directions, because most waves anyway propagate in one major direction. So across all of these analyses, what the picture that emerges is, I think, you have a macroscale wave, which you can be thought of as a wave of oscillations. But then if you look at each individual local patch of cortex, it likely, when there is an excitable phase of this macro oscillation, you get some spiking, and that in turn can trigger some waves based on previous findings that spikes can possibly trigger microscale waves. And we've also shown that these waves in turn can recruit more spiking, but although in a sparse way. Right? So think of this as an injection of current into a local network of neurons, which then creates some deflections in the field that then propagate as traveling waves. And during this propagation, it further depolarizes the local network, and you see an increased probability of spiking as the wave propagates at the microscale. Okay, so you induce sparse waves with that in turn weakly modulates further spiking. But of course, we should also notice that these microscale waves can contribute back to the macro LFP. After all, we are talking about a complex system where there are no clear one-way chains of causation, right? So these all influence each other ultimately. And so rather than these verbal descriptions, we should really be modeling these things in detail to understand how such coordination arises. So to tie the pieces together, we have macroscale waves that propagate over these long-range myelinated fibers, and spikes or bursts might be transmitted to other areas, inducing changes to the field potentials there. And if you have excitatory phases there, it might in turn trigger some sparse spiking. Now, of course, the sparse spiking might trigger local waves, which we currently do not have direct evidence for, but there are suggestions in the literature that this is the case. But we have also shown now that these waves can in turn recruit more spiking along the way. And we've also shown that these waves do induce repeatable phase relationships between electrodes, some falling within STDP windows. And these LFP waves in turn weakly modulate spiking. And the degree to which these traveling waves occur in the lateral temporal cortex, and especially in the anterior, so I haven't shown you these differences between anterior and posterior, but it is especially in the anterior temporal lobe that the rates of occurrence of these waves are strongly indicative of memory performance, but at the subject level. So these suggest that traveling waves can provide these windows of learning and STDP. So think of this as a background of opportunities for learning. And so if there is stimulus-specific representations and spiking that come on, they can leverage these windows that have been set up by these traveling waves, making it more likely that you encode things successfully. Okay, so with that, I would like to also briefly mention some ongoing work with complex waves, because we know that it's not just planar traveling waves that occur. We also have more complex wave patterns. So this is work that is being done by a research associate in my lab, Arunik Biswas. By the way, he's looking for a PhD position, and he's built up a lot of expertise in wave analyses, but also multi-level mixed effects modeling and such. And I apologize for this advertisement, but you all probably know how difficult it is for international students these days. And I think this is a very prepared candidate if you're looking for a PhD position, PhD candidate. So there are a couple of different approaches you can use to detect complex wave patterns in these kinds of LFP recordings. So one approach is you can detect critical points in the velocity fields, which are these apparent points of zero velocity. And you can assume linearity around these critical points. And there are established methods that allow you to automatically classify activity around the critical points as spiral type activity, as source, sink type activity, and so on. But I think Earl Miller's lab also uses another approach where they directly fit phase rotation maps, but using the phases. Arunik has used a similar approach, except that he uses the velocity fields themselves to detect spiral waves. And so you see pretty nice rotating waves also in these at these Utah arrays, right? So you can see if you follow any color along, it kind of rotates around. And so Earl Miller has shown that in monkeys doing working memory tasks, if you look at the prefrontal cortex, you see a greater number of spiral compared to planar traveling waves. But we see the opposite. We see a greater number of planar traveling waves compared to spirals. And this is just to show you that we've been very careful to separate out the planar traveling waves, which were higher than zero point 0.7 if you look at the phase gradient directionality. But if you look at these spiral waves, they take on all of these PGD values. But the important finding so far, at least by looking at a couple of participants so far, is that we see even greater spike locking if you look at spirals compared to planar traveling waves. So these are the same results that I showed you earlier, where you see weak modulation of spiking activity if you trigger spikes to the onset of planar traveling waves. But if you look at spirals, that relationship is even stronger. And you see that across different sessions of the same participant, but we've also seen it now for a second participant. So what all of this suggests is that both planar but also other complex propagating wave patterns weakly modulate spiking, possibly setting up STDP windows for different combinations of electrodes and sites depending on the propagation pattern. And we've seen that the rate of occurrence of these waves, which coordinate local waves and spiking, is indicative of overall cognitive performance. Okay, so with that, I'd like to thank the organizers and Kareem, of course. This is where I first got introduced to this idea of traveling waves. We've spent a lot of time looking at these movies, and all the members of Kareem's lab, as well as my own lab now, for helping out with various aspects of this research. Okay, and thank you.
Vishnu, that was wonderful. Thank you. Yes,
Yeah,
clapping
thanks.
for clapping. Um, why don't we open the field to take some questions?
While people start putting questions in the chat, I'll just ask something about one of your last points. Uh, why do you think spiral waves might be more linked, or more strongly linked to spiking than the planar waves?
Yeah, it's a good question. I think ultimately the answer will probably come from detailed modeling of these things. This is fairly new, I think. So these analyses that I showed you at the very end are literally 1 week old. So we are still trying to make sense of these results. I think in the anesthetized marmosets, they saw that some of these complex waves are associated with spiking to a greater degree than planar traveling waves. So I think that's the 2015 J Neuroscience paper. I'm glad actually that you all have Pulin Gong coming up next because that's work from his lab. So there are some indications already that spiking is probably more coordinated by these more complex wave patterns. But why? I'm not sure.
Thanks.
I had a question also. I'm just trying to reconcile the macros. I mean, do you believe that the macroscopic waves are generated by the same processes as the microscopic waves? And you're just seeing a blow up of it, or is it completely different process and one is modulating the other? That the microscopic waves are more related to spiking, and the macroscopic waves are just some sort of ongoing excitability.
Right. So I think to answer this question, at least in the context of the sort of task that we looked at, I guess we'll have to go to the visual cortex first and think about what induced probably some oscillations there that in turn could have propagated via these Watt pathways, right, over to the temporal lateral temporal cortex. And since we are also talking about alpha, I don't think we quite know what the generators of alpha are exactly. It could be thalamocortical, it could be entirely cortical. So if you look at the 2019 Halgren paper, they suggest that it could be entirely cortical in origin. So ultimately, I mean, we do have models that suggest how these oscillations come about, right? But once these oscillations come about, of course, work by several people, including you yourself, have shown that if you set up these weakly coupled oscillator systems, you get these macroscopic waves, right? But like I also indicated in one of my slides, I think that the kind of modeling that we require to answer this question really is the kind of modeling that Mueller did, which is they started with these integrate and fire neurons. They summed over the activities across these neurons to simulate the LFP. But I think we've got to build that all the way to the ECOG scale to really try and understand the bidirectional connections between these scales. And without having done this modeling work, it's really very difficult to speculate about the direction of influence of these things. And I've played around a little bit. So I've been playing around with, let's say, a Kuramoto oscillator system at the macro scale, Wilson Cowan at the micro scale, because I wanted some EI stuff going on at the micro scale. So I mean, we could start off with that kind of a system to try and understand how these things work. But ultimately, I think we also need the single neuron stuff to be modeled simultaneously. But yes, so I don't have a good answer for what the direction of causation should be, if at all there is such a direction. But I think this sort of modeling needs to happen.
Maybe related to that, Vishnu. By the way, great talk. Why do you think?
Thanks.
Well, I didn't quite got the relation to STDP. Do you think it's exclusively STDP, or you mean any spike field type of interaction?
Yeah, so it's just that if you look at various pairs of electrodes during these waves, and if you look at the phase relationship between them, you can compute an associated time delay between them. They fall within this, 20, 30 milliseconds that people consider import for STDP. And that's a similar calculation that Mueller did, suggesting that during these waves, about 50% of electrode pairs are within this window, right? So if you set up these windows, then if you have task-specific activations that come up that can leverage these windows that have been set up, then it means that you should observe greater chances of encoding during these windows. Okay, but yes, of course, this is still very much speculative.
It could be BTSP, or I don't know what time intervals in the. Is it during the task, or is it during the if it's spontaneous activity? Maybe that's a strong.
So you mean the wave rates that I observed and the relationship to
Yep.
the memory performance? Yeah, it doesn't seem to matter where I pick the waves from to do that calculation. It just seems that in these more optimized brains, it seems like there are more waves in general. So which is why I said that I could pick up just the incorrect trials. I could pick up just the baseline periods, and it seems to work. It seems to show the same kind of pattern.
It's irrelevant to behavior, yeah.
Sorry?
So it's irrelevant to behavior, yeah.
Well, it's relevant to behavior in the sense that it is predictive of overall performance, right? But if you're asking me about whether it distinguishes between successful memory and unsuccessful memory, I don't see that. However, there is this caveat that I have no frequency precision in my analysis. So perhaps the waves that matter for that kind of differentiation, right, between memory conditions could be very frequency specific. And yeah, so I don't have that analysis because I was very focused on this temporal precision.
Thank you.
Yeah.
Vishnu, that issue of frequency you just mentioned is interesting because so many analyses of traveling waves think about frequency a lot, and you are not, and I get it. But I wonder if I'm trying to understand more about the waves that do and do not match up between the micro and macro waves, and I wonder if frequency would provide some useful information, especially because of issues related to aliasing, which is something Irfan once mentioned. I don't know. I'm just trying to understand that more.
Yeah, so what I can show you perhaps is yeah, well, we
By
don't
the way,
have to go. Yeah.
while you look through your slides, I wanted to encourage everybody else in the audience. It's really great that myself and Irfan and Bard and Uma have lots of questions, but if you have questions in the audience, please jump in or put your question in the chat and we can call you up.
Yeah, so what I'm showing you here is something that everybody understands, which is that, so I simulated some sinusoids with some noise infused, and you can apply different kinds of filters on the data, right? So in the red, you see a very narrow band filter. right? And in the yellow, you see a wider band filter. And you can see that, yes, this is the period where you have this oscillation. But if you look at the reds, you do extend the oscillations, even though there are no actual oscillations here in time. And that is just a natural consequence of demanding great precision in frequency. So you extend the cycles in time, right? So while we understand that, I mean, this is a question essentially, right? So why are your waves so transient? Because other analyses in the literature report multiple cycles of these waves. And I think that is related to the frequency filtering that I just showed you on the previous slide. And my response to this is maybe they actually are transient. Because if you demand frequency precision, then naturally we don't have a precise estimate of the duration of the waves, right? Maybe the mixing of frequencies in my analysis is obscuring some longer waves. That is also a possibility, right? But I can get you long multi-cycle waves if I simply process my wave differently with a 3 hertz band around the coherence frequency, which a lot of people do in the literature, right? So for example, this is a wave that I detect using such a narrow band filter. So you can see 4 or 5 cycles of the wave that appear. So any questions about durations, any questions about transients can only be answered in the context of the signal processing that has been done in the pre-processing, right? And so that is something that I think we all should keep in mind when we try to ask and answer such questions, right? Um, And there are some results in the old preprint. You can even see directionality effects from baseline to task and all that if you look at frequency specific waves. But you don't see that if you do this very temporally precise analysis, right? So I think you might need different motivations for frequency filtering when it comes to these different questions.
Questions?
Well, I have another one too.
Okay.
This is a point when you're dealing with oscillatory waves. I I often argue with other people about this, about whether it even makes sense to talk about velocity. Because if you've got two synchronous guys, the velocity's infinite, right? And any little phase shift is going to make that velocity vary a great deal. And it's nothing to do with any passage through fibers or anything. It's just when you measure velocity in an oscillatory wave, you're just looking at the phase difference divided by the frequency or times the frequency, right? So I always wonder when people talk about velocity of these oscillatory waves, why they focus so much on velocity, because it really um, if this guy and this guy go together, the velocity was infinite, right?
Mhm.
So when you're talking about phase waves, which is what you're measuring here, right? Getting the analytic phase and you're getting a phase. I just wanted to say I never think it makes that much sense to talk about velocity. That's all.
Okay. Uh, yeah, I'm not sure I actually quite understand the question.
It, it was
So yes, if you have complete synchrony, yes, the velocity is infinite, but I didn't quite understand why it doesn't make sense to compute the rate at which phase changes across space and time.
it makes sense, but it really depends on what the frequent underlying frequency is. The underlying frequency, the phase difference times the frequency kind of gives you the velocity. Um, and so the velocity doesn't depend on transmission velocity across fibers because that's not frequency dependent. That's a fixed thing. And your phase velocity that you measure is completely frequency dependent. So if I have a certain pi over 2 phase difference and I'm going at alpha versus gamma, we're talking about a huge difference in velocity. That's
Yeah,
what
point taken. Yes, yes, understood. So it's really the oscillatory dynamics of the nodes and how they are coupled and all that leads to the velocities is what you're saying.
yeah.
Yeah, right.
I
Yeah,
think what
so
Bart
I
is
guess,
saying is that
yeah,
apparent is that you might call something velocity, but really it's a really high velocity, but really it's just a small phase shift because
right.
mathematically they're
Yeah,
the same thing, but
yeah,
physiologically they're super different. I think
yeah,
that's what.
makes sense. Yep.
Vishnu, I have a quick question about one of your newer results about wave prevalence being predictive of subject performance across
Mhm.
subjects. I was wondering if you're seeing that things like how long range those waves are or how bidirectional those waves are um, also have any predictive uh, influence in overall performance.
Yeah, so that's a good question because I thought of doing this by separating out the waves in terms of which direction they go in. But because of the extreme directional preference, you see like 80% waves that propagate in the posterior to anterior direction compared to the other. So it's usually not enough data in the other direction to compute these things robustly. But I should try it nevertheless. So the answer is I haven't actually looked at it yet. Right.
Sure.
So it's, yeah, like you said, it's a newer result. But I was thinking that the balance is so shifted in favor of one direction over the other that it's unlikely to yield very big differences. But we'll see. Yeah.
Cool. Thank you. Are there any other questions? Okay, if not, thank you, Vishnu. This was awesome. Oh, we do have one more. Um,
See some chats.
Well, the question is, Zoom user, if you would like, you can ask your own question to Vishnu and talk, or we're happy to read it for you. Or Vishnu can read it himself.
Let's see. I have to. Oh,
The question
I've
is there any
lost.
underlying biological reason why waves travel in one particular direction versus another?
So the models that generate these would have to assume anisotropies in the in the connectivity profiles, right? So I would assume that something like this is at play. But I think work from Josh's lab and also these general ways of modeling these things have also shown that there could be intrinsic frequency gradients that could also explain directionalities, right? So it could be a combination of these factors that explain why we have directional preference.
Okay. Anybody else?
Mm.
Vishnu, I think it's really cool work, and there's nobody else that I'm aware of looking at these different kinds of waves. Um, there are a lot of, and I have a lot of questions, and it's a kind of work that opens up new directions. So I'm happy you're doing it. I'm having your, it seems after a little gap, you're continuing
Yeah.
with the work, and that makes me happy. So thank you.
All right. Thanks
And
a
thanks for
lot,
your
Josh.
talk, and,
Yeah.
and thanks for your talk today too.
Thank you very much. Yeah. I had a lot of fun, and thanks again for inviting me to do this. All right.
Yeah. Good talk.
Yeah.
Thank you.